Thank you, ma'am. After a wonderful talk on genetics in retinal diseases, we'll go on to how ocular genetics involves corneal disorders, and I'll be briefly presenting where we formed a national consortium for genetic diseases. As we all know, ocular genetics in corneal disorders have been largely revolving around corneal dystrophies, keratoconus, and anterior segment dysgenesis. Corneal dystrophy has been largely classified only on the basis of genetic knowledge that we have had, and the current as well as the first version of the International Committee for Classification of Corneal Dystrophies was based on a genetic classification where there were 4 categories of corneal dystrophies depending on whether the loci or on the chromosome were mapped or whether the proper gene was mapped or not. So category 1 corneal dystrophies are those where we know the gene and the locus has already been mapped, and a well-defined corneal dystrophy like the TGF-beta1 dystrophy is category 1 corneal dystrophy. Category 2 dystrophies are well-defined dystrophies where the chromosome locus is known, but the gene has not yet been identified and the mutations are still being, the research is still ongoing. Category 3 corneal dystrophies are well defined phenotypically, but the gene and as well as the chromosome locus is not known. And all other dystrophies which have been described but where the genetic hereditary pattern is not yet known, or whether they are really corneal dystrophies or degenerations, they still form an unclassified group, are category 4 corneal dystrophies, and this has been retained in the 2015 version of the IC3D. Coming on to anterior segment dysgenesis, which is an ill-defined group of developmental abnormalities which usually involve the cornea, the iris, and sometimes the lens. This classification is largely based on the phenotype now, but in future we hope that a genetic classification will be there. But since it involves multiple genes and it's not a single gene disorder, we are hoping for a good genetic classification for the anterior segment dysgenesis as well. So just to go on the evolution of anterior segment dysgenesis, the current knowledge is we have defined anterior segment dysgenesis on the pattern of the embryonal development and on the neural crest development, or whether they are from the non-neural crest origin, whether it comes from the surface or the neuroectoderm. So we have a multitude of anterior segment dysgenesis which can range from megalocornea to Axenfeld Rieger's to aniridia, and depending on which morphological phenotype the patients fall into, they are classified according to, and we do have a genotype or a locus which has been identified, but it is not particularly, some of the genes have been mapped for all these disorders. So just to bring all of us together on the national front, we had a low vision workshop where we got together in September 2015 to work out and form a national consortium of ocular genetics, and it was very appreciated, and it was involved a lot of experts from which involved ophthalmologists, pediatricians and geneticists from all over the country, from Delhi and Shankar Nethralaya, Narayan Nethralaya, Aditya Jyoti Institute and Srikaran Institute. So this was the deliberations that happened during the national consortium. It was it was recommended that we'll have a national registry for ophthalmic genetic disorders, and a meta-analysis of all the pooled genetic data in the country will be analyzed. And it was also proposed that a society which will be known as SOGOOD, Society for Study of Genetics for Ophthalmic Disorders, will be formed. So then a brainstorming session was thought of doing a collaborative research, and then we thought we'll focus on a single gene disorder first and then go on to multiple gene disorders. And so a consortium worked on basically these 4 diseases in the anterior segment, which involve the Fuchs endothelial corneal dystrophy, congenital hereditary endothelial dystrophy, and anterior segment dysgenesis. Going by the previous project that was done under Professor Radhika Tandon from RP Center, wherein for 300 patients were screened, 300 patients and family members were screened. So it involved around 550 individuals. We did get some genes linked to the stromal dystrophies and the endothelial dystrophies. So TGF beta 1 was found in the granular corneal dystrophy, CHST6 in macular corneal dystrophy, and in CHED we had the solute carrier for A11, and in the Fuchs we had a multitude of variants from SLC4A11 to TCF4 and zinc finger box gene. These were the various publications that came out of that project, and which worked on all the endothelial dystrophies as well as the avellino and the granular and the macular corneal dystrophy. So as part of the national consortium, we did develop an Indo-US collaboration for in the first phase for CHED and Fuchs dystrophy. This was being funded by the National Institute of Health from the US and the Department of Biotechnology from India. So in CHED, the participating centers were the ophthalmic department and the genetics department of AIIMS, New Delhi, LV Prasad Eye Institute, Hyderabad, Arvind from Madurai, and Jules Steen Eye Institute in US. The specific aims of studying CHED was to find individuals who did not have any solute carrier for mutations, to find what is the genetic basis of these through newer techniques, through whole exome sequencing and comparative genomic hybridization, and to find out whether this SLC4A11 mutation, what proteins are affected, and whether it does affect the human endothelial function, and what effect it has on the phenotype of CHED. And the second disease, the corneal disease that was proposed to be studied, was Fuchs endothelial corneal dystrophy, and the participating centers here were AIIMS, Arvind, and Sri Kiran Institute of Ophthalmology in Andhra Pradesh, with collaborating with Texas and United States. In this, we plan to determine the prevalence of TCF4 and the ZEBI polymorphisms in Fuchs corneal dystrophy. And again, we needed to see the protein expression in donor corneas and develop models through this. So the way forward would be that we have already targeted on the CHED and Fuchs dystrophy. Since we thought what would be the scope of novelty in studying anterior segment mesodermal dysgenesis, so we thought we'd work on the genotype and correlate the phenotype with it to form a classification scheme which would be based on the genetic classification. Thank you.